Hi, my name is Eman Toraih. I'm a research fellow in biotech. I am an assistant research professor in the Department of Surgery at Tulane University. Thank you for joining me today as I share the findings of novel markers for predicting malignancy in thyroid nodules. This study offers a better modality for cancer detection that's more conclusive and fits all racial groups. Thyroid cancer is a common type of endocrine malignancy. Its incidence has been steadily increasing in many regions of the world. The vast majority of thyroid tumors arise from thyroid follicular epithelial cells, where 3 to 5% of cancers originate from parafollicular cells. The follicular cell-derived cancers are further subdivided into three categories: well-differentiated papillary and follicular carcinomas, poorly differentiated carcinomas, and anaplastic or undifferentiated carcinoma. Follicular adenoma is a benign tumor that may serve as a precursor for some follicular carcinomas. In patients presenting with thyroid nodules, the clinical challenge is to accurately and rapidly identify those nodules that harbor cancer. Similar to other cancer types, thyroid cancer initiation and progression occurs through gradual accumulation of various genetic and epigenetic alterations. This includes somatic mutations and microRNA dysregulation. Thyroid cancer represents a type of neoplasia in which critical genes are frequently mutated via two distinct molecular mechanisms: point mutation of particular genes or chromosomal rearrangements at particular sites. Fine needle aspiration is used to evaluate thyroid nodules and remains the worldwide gold standard for workup. Fine needle aspiration results are useful for stratifying malignancy risk. However, only looking at the cytopathology is challenging in some cases, and further molecular tests are proceeded, such as mutation screening and microRNA suppression analysis. These tests are essential to provide key information for counseling patients and determining the appropriateness of surgery. The minority population in the U.S. accounts for 27.6% of the population. So looking into the SEER database, we found out population with African American descent had the least incidence rate. However, they had the worst 5-year survival, highlighting the possibility of being unreported or underdiagnosed. Here at Tulane, on analyzing 263 patients at Tulane Hospital who had molecular diagnostic tests and underwent surgical removal of the nodule, we found out through our clinical practice that 50 to 67% who were misdiagnosed are non-white, leading to unnecessary surgeries and complications or two-step surgical management, thus highlighting the urgent need to investigate the possibility of the concept that not all tests fit all. Therefore, we aimed to identify and validate a microRNA signature across racial groups to diagnose cancer in patients presenting with thyroid nodules. This is to test that if African Americans, Asians, and Hispanics require different molecular biomarkers than white non-Hispanics. In addition, using both RNA and microRNA transcriptomic data, we aimed to identify the driver mechanisms of thyroid tumorigenesis in each race using the public repository's TCGA database. Our analysis included 9 steps. We downloaded the required data for each racial group separately: Black, Asian, Hispanic, and white non-Hispanic. Then we did exploratory analysis to visualize the variance among the data samples. Box plots were first generated to represent the sum of all genes within each sample. Then principal component analysis, or PCA, was performed in 4 racial subgroups to look at the subgrouping pattern of cancer and controls and detect outliers among the samples. Then, using the microRNA sequencing data, comparative analysis was performed within each racial strata to compare between cancer versus non-cancer samples by running the differential expression pipeline. Look-to counts per million of the data was used, and HR method was applied. Significance was set if the folded change exceeds plus or minus 1 and adjusted p-value below 0.05. And downregulated and upregulated genes were visualized in these 4 types of plots: volcano plot, Venn diagram, upset plot, and cluster analysis. Ward D2 linkage and Euclidean distance measures were used in the hierarchical clustering analysis. In step 4, we performed supervised machine learning technique and evaluation matrix to construct a model for predicting malignancy in thyroid nodules. Decision tree for classification was employed, which yielded triple microRNA biomarkers with the best discriminator ability. For clinical application, we generated a diagnostic nomogram to visualize the results of logistic regression. A risk score was generated from beta coefficient of logistic regression model and evaluated by receiver operator characteristic curve or ROC curve analysis. Prediction performance was compared to the current molecular microRNA panel that's currently used in the clinic. For validation, stratified analysis of the TCGE data by demographic and pathological characteristics, including age, sex, disease stage, and histopathological variants, were performed to compare the performance of the model in each state. The expression pattern of the final 3 microRNAs were also investigated in other independent datasets in the Gene Expression Omnibus database for external validation. Furthermore, we carried out function enrichment analysis to unleash the biological context of the 3 microRNAs, which fits all the racial groups. On the other hand, to identify the driver mechanisms specific for thyroid cancer in each race, we performed RNA sequencing analysis. We concatenated log-normalized the RNA and microRNA expression matrices together, intersected the differentially expressed genes and differentially expressed microRNAs with gene targets identified from prediction tools and experimentally validated databases. Then the final lists were integrated to build a microRNA gene network. Next, to analyze the tumor microenvironment, we assessed the tumor purity and the composition of immune cell infiltration using 3 software: Estimate, CyberSort, and microenvironment cell population counter methods. Next, Upstream regulatory network was constructed through analysis of the transcription factor targets derived from the Jasper and ChIP-seq database. And kinase enrichment analysis was performed using the X2K online database. Function enrichment analysis for the up and down regulated genes and microRNAs were explored for each racial group. And this to identify the driving pathways and gene ontology annotation is specific for each cohort. Transcriptomic signature of 495 thyroid cancer and 59 non-cancer tissue samples were retrieved from the TCGA database. In summary, over 2,500 microRNAs and over 20,000 messenger RNAs and long non-coding RNAs from RNA-seq and microRNA-seq were included. Clinical, pathological, and molecular information were obtained from the CBio portal of Cancer, Genomic, and FireBrowse. To gain insight about the dataset as a whole, we performed dimensionality reduction using a principal component analysis for each racial groups. We reduced the data of all samples into 2 dimensions that best represent our data. In the 4PCA plot, there was good separation between cancer and control samples with very few overlap in the white non-Hispanic group cohorts. Volcanoplots was performed to identify differentially expressed microRNAs in each racial group. The plot is showing the log2 folded change along the x-axis and negative log10 of the adjusted p-value along the y-axis. Each dot represents 1 microRNA. At the cutoff of absolute value of log fold at 1 and the Q below 0.05, significant genes are defined in blue for downregulated and red for upregulated microRNAs. That yields significant 26 microRNAs in Black, 28 in Asians, 29 in Hispanics, and 81 in white non-Hispanics. As demonstrated in the Venn diagram, intersection between the 4 racial groups showed common 11 microRNAs that were significantly deregulated. 10 were overexpressed, and the last one was downregulated. Currently, the standard of care relies on 11 microRNA panels used to distinguish between malignant and benign thyroid nodules, as demonstrated in the first horizontal bar. In the 2 blue boxes that enclose 7 and 4 microRNAs, these are our 11 microRNAs that are consistently deregulated in all racial groups from the TCGA data. As marked by the yellow arrow, only 4 microRNAs of these existed in the currently used molecular testing. Some other microRNAs don't have a role at all in any of the races. This heat map shows the expression level of microRNAs across the racial groups. The new panel of 11 microRNAs, one of them were downregulated in blue and all others were upregulated. On the other hand, the old panel included 11 microRNAs. 4 microRNAs were in common with our new panel. 5 of them did not show significance in one or more of the racial groups, highlighting its low potential to have good diagnostic performance for all thyroid cancer patients, especially non-white patients. In the hierarchical clustering analysis, the algorithm detects closest objects with similar pattern and links them together in the left photo. Our data-driven clusters showed that control samples in orange were assigned together, having less distance between the samples, and forms distinct cluster from cancer patients. Unlike the old panel in the right, normal tissues were split across different clusters. Predictive model using decision tree, 3 microRNA biomarkers were revealed to be the best discriminator for malignancy. The decision tree plot has nodes connected by edges. The best classifier feature is at the root node at the top, which was microRNA 34A. The height of this decision tree is 3. At the bottommost level, we have the leaf nodes that hold the output label, cancer or normal. With a combination of 2 other microRNAs, 144 and 146p, at a cutoff around 5-fold change, we had the best diagnostic ability. Next, logistic nomogram was generated using the demographic characteristics at the time of presentation and the expression profile of the 3 microRNAs. It consists of several horizontal lines marked off to scale. For each of the 6 parameters, it gives a point score. Their sum predicts the risk of malignancy in patients with thyroid nodules. So here in this example, this 60-year-old white non-Hispanic male patient have 285 total points, which is equivalent to 81.6% risk of malignancy. So to calculate the cumulative genetic risk of cancer, a risk score was generated using the 3 microRNAs. Multivariate logistic regression analysis was performed, and the beta coefficient of the regression model was used for imputation, and using the formula of the sum of the product of microRNA expression and its beta coefficient for each sample. Receiver operator characteristic curve analysis was used to describe the diagnostic ability of our new microRNA risk score and the accuracy of the currently used microRNA panel. The closer the curve towards the upper left corner, the better the prediction ability of the diagnostic index is. As demonstrated here, the area under the curve, or AUC, of our generated risk score shows a perfect prediction ability with 94% sensitivity and 98% specificity, while the old panel had poor predictive value to discriminate between malignant and benign nodules. On the right, kernel density plot shows higher range of scores in cancer samples compared to normal samples. To test its efficiency at different disease conditions, iteration of analysis using data split by sex, age, subtype, stage, etc., the ability of the microRNA risk score to accurately predict malignancy was assessed using the concordance C statistic. Here, as we can see, the risk score showed excellent discrimination in all conditions of patients. We analyzed 2 other datasets, and we had similar results for microRNA 34 and 146b were upregulated in cancer, while 144 were downregulated compared to controls. In the first experiment, 5 paired samples, tissue samples of papillary thyroid carcinoma and non-cancer tissues of the same individuals were analyzed, and using the same formula, risk score was estimated from the 3 microRNAs. Similar to our primary findings, high score was associated with malignancy. In the other experiment, 8 pairs of tissue samples were analyzed, yielded similar results. The same 3 markers are frequently deregulated in other cancers as well. Enrichment analysis revealed significant enrollment of microRNA 34, in particular in thyroid cancer. as shown in red box, and was involved in other cancer types in blue boxes. They were enriched in essential cancer-related pathways as adherent junction, B53 signaling pathway, and cell cycle pathway. MicroRNAs targets 16 genes within thyroid cancer KEGG pathway at different stages of thyroid carcinogenesis, from normal to PTC, from normal to adenoma to follicular thyroid cancer, and to undifferentiated carcinoma, thus highlighting the role in cancer development at different subtypes of thyroid cancer. The top most significant gene ontology terms were related to cell cycle, response to stress, protein assembly, chromatin urbanization, and cell death. All these processes are closely related to thyroid cancer tumorigenesis in the literature. Other molecular function terms were related to cytoskeleton and methylation. To construct a gene microRNA regulatory network, RNA-seq for each race was performed through which the differentially expressed genes were defined. Here, this Venn diagram shows the differentially expressed genes in each racial group. Intersection demonstrated a common 609 genes that are deregulated in all cohorts. Co-expression analysis was performed for each racial groups. Those pairs with correlation coefficient of less than -0.5 were selected. Gene targets were identified from multiple well-annotated databases, and results were intersected with the correlated pairs. A final of 73 microRNA gene target interactions were further explored. Regarding the tumor microenvironment, we used estimate software. It's a tool to calculate the fraction of tumor cells in clinical tumor samples using the next generation sequencing data. So we used the TCGA data, and we found no significant difference in the distribution and comparisons of stromal and immune scores between different racial groups. Distribution and comparisons of stroma and immune scores among different tumor stages, differentiation grades, metastasis, histological subtypes, and recurrence also showed no significant difference as well. Regarding quantification of the exact infiltrating immune cells, we used two softwares, CyberSort and MCP counter methods. A CyberSort tool was used to estimate and distinguish 22 commonly infiltrating immune cells in thyroid cancer patients with different race, while microenvironment cell population counter method quantified the absolute abundance of 8 immune and 2 stromal cell populations in heterogeneous tissues from using gene expression data. Transcriptomic profiles of thyroid cancer samples were used to infer tumor cellularity as well as different infiltrating normal cells. We found out that mast cell activated immune cell was significantly higher in white non-Hispanic population than white Hispanics. To identify the driving mechanism for each racial groups, microRNA and gene set enrichment analysis was performed. KEGG pathway analysis showed 3 common pathways enriched for the 4 groups, and these includes transcriptional misregulation in cancer and extracellular matrix receptor interaction. In Black population, differentially expressed genes and differentially expressed microRNAs were enriched in axon guidance and retinol metabolism, which is similar to the literature. Asian population, genes were more related to several autoimmune disorders and immune-related pathways, while in Hispanics, B53 signaling pathway was significant. The well-known pathway is related to BRAF mutation as CAMP, PI3K ACT, or MAPK signaling pathways were enriched in white non-Hispanic dataset. Transcription factor targets were explored based on the gene expression data. We found 89 transcription factors to be enriched. The top 20 are shown. Some of the genes, like FOXY1 and GETA2, can target hundreds of differentially expressed genes. two of the significantly enriched transcription factor list were differentially expressed in our cohorts. ChIP-seq enrichment analysis prioritized transcription factors that most likely to regulate the observed changes in messenger RNA expression in each race. Some of these were up or down regulated in our thyroid cancer samples. Kinase enrichment analysis was performed to prioritize protein kinases known to phosphorylate substrates within the subnetwork of transcription factors and the connecting proteins. As shown here, the top kinases that are enriched using the specific differentially expressed genes of each group. Intersecting the significant kinase list with the corresponding differentially expressed genes in each race defined the upregulated kinases within thyroid cancer dataset. We used that list of upregulated kinases to find kinase inhibitors or drug targets using the Kinase MD database. As shown, anaplastic lymphoma kinase receptor or ALK was common in all groups. ALK tyrosine kinase inhibitor TAE684 drug was shown to have anti-tumor activity in 4 thyroid cancer cell lines. During the current analysis, we performed microRNA and RNA sequencing analysis in thyroid cancer tissue samples. Based on our TCGA analysis, we identified differentially expressed genes and differentially expressed microRNAs. The enrichment showed that tumorigenesis process driving thyroid cancer development was different in each racial group. We also developed and validated a diagnostic molecular test of 3 microRNAs, which seems to have better accuracy in white and non-white population. Adding our markers as ancillary tests to the current standard of care might have a greater impact in improving diagnostics in thyroid nodules across all racial groups. At this end, I'm grateful that I was working with the Omics Logic team under the mentorship of Drs. Raghav, Kaur, and Ilya. I had a chance to explore and extract meaningful insights from omics data that will expand my career opportunities. Please, if you have any questions, suggestion, or feedback, please reach out to me through my email, etoraih@tulane.edu. Thank you.